I'm Sarah Halter. I'm the Executive Director of the Indiana Medical History Museum, and I'm very excited to see a lot of new faces here today. This event is one that we've been really excited about and looking forward to. And I know that all of you have too. So I appreciate you joining us tonight, whether you're here in the old pathology building or joining us online. This is the fall 2025 installment of the Forensic Science Lecture Series. A lot of you, I think, are familiar with that series. This is a very long time partnership between the Indiana Medical History Museum and the University of Indianapolis Human Identification Laboratory. That's not what it was called back in 2006 when we began this series, but that's what it is now. We are very grateful to Dr. Goodpaster for being here today to share his work with us, and we're all really excited to hear about it. The title alone, I think, really sucks you in. Dr. John Goodpaster received his BA in chemistry from Gustavus Adolphus College in St. Peter, Minnesota. He then he attended graduate school at Michigan State University, where he earned an MS in criminal justice and a PhD in analytical chemistry. Following postdoctoral studies at the National Institute of Standards and Technology in Maryland, he served as a forensic chemist with the Bureau of Alcohol, Tobacco, and Firearms and Explosives. Dr. Goodpaster is now a professor in the Department of Chemistry and Chemical Biology at Indiana University, Indianapolis. He's also part of the Forensic and Investigative Sciences program at IUI. He teaches in the areas of alcohol and drug analysis, as well as trace evidence. Ongoing research projects in Dr. Goodpaster's laboratory include chemical analysis of human hair, coupling gas chromatography and vacuum UV spectroscopy, and I have no idea what that means, so if you want to know, you'll have to ask. Profiling of drugs of abuse using chemometric techniques, is that how you say it? To classify and associate trace evidence, and also studying and training and performance of explosive detective canines. It's very, very exciting over there. Please help me welcome Dr. Goodpaster.
All right. Well, good evening. I'm really pleased to be here. I've been here before, just coming through as a guest. I think this place is fascinating. I love staring at brains in jars. I think that's pretty intriguing. It's really a lot of fun to be here tonight to talk about this particular case that I got involved with. The basic story that I'm going to try to tell is that chemical compounds are all around us all the time. And even though we cannot see them, we can sense them through our nose. Okay? You're walking around essentially with a chemical sensor on your face. And in particular, we know enough at this point about these volatile compounds so we can associate them with their source. So we know, for example, and this is really relevant here, we know what kind of volatile compounds come out of you when you die. Okay, so the moment you die, everything stops, all your living processes stop, but microbiological processes begin. And that whole process of decay and decomposition is something that people spend a lot of time and energy studying. And so we actually know a fair amount about it. And so what happened is my laboratory, we study a number of different things that you heard, but we definitely study volatile compounds, things that are easily evaporated. We study how canines can detect those compounds, and we're particularly interested in explosives. But then this case came along because they wanted us to find out whether or not we could see or identify any volatile compounds that are associated with decomposition, even though the body was long gone years ago. It ended up being a pretty interesting study. We ended up developing decent data. I testified in this case, and the case has now been fully adjudicated, so I can sort of lay it all out for you. Okay? So, the scent of death. Do I need to do that reconnection thing you just told me? There. Yeah, I should have made us skip this one because that's been covered. So I'm going to begin with just acknowledgments because it was definitely not just me doing this. This case came out of Michigan City, Indiana. The detective in charge is right there. That's Anna Painter. She's now a lieutenant there. And she was the one that approached us, explained the background of the case, and asked us to see if we could help. At IUI, we are, if you are not aware of it, Indianapolis is actually a pretty well recognized center for forensic science in the United States. And the forensic science program at IUI has been around for about 20 years. And this is a picture of myself and my graduate students. Alexis Heckert came out of the forensic science program at IUI and now has stayed, and she's doing her PhD in chemistry. She's right there at the bottom, right there. So she actually was directly involved with processing samples. And I want to make sure I acknowledge her efforts in this. Okay, so this is the agenda. We're going to talk about the case itself, the murder, talk about sense of death in detail, and then what we actually did, the measurements we made, and then end with just sort of some general comments about why VOCs can be so important. Okay, so the victim's name is John Gonzalez. And I say it's not exactly a whodunit because in this case, someone confessed. So the murder occurred many years past. The defendant then confessed, but then recanted. Okay, so you might think, well, geez, if somebody confesses to a crime, why do we even have to do anything, right? Just throw them in jail. Well, as we've learned, confessions are not always 100% reliable. Now, his particular confession, to be fair, I guess it's not really fair to him, but his particular confession is extremely detailed. So it's pretty obvious he knew what he was talking about. But just because someone says they did something doesn't mean they did. And so it helps and it's important to have additional evidence. And obviously, in this case, he changed his mind. I would wager he probably changed his mind once he figured out what the jail time is going to be for homicide in Indiana. And so he said, no, I was just kidding. I didn't do anything. Okay. So then, we come along and we try to, put in more of the pieces. And so obviously the detective was heavily involved in this case, tracking down the victim, looking for traces or evidence of when that victim was last alive, accessing their bank accounts and being seen by people, all that kind of good stuff. And then the crime scene itself turned out to be a basement. And that's where the body was actually stored. That's Michigan City, Indiana, way there at the top. And so right on, literally right on the Lake Michigan. So beautiful place. It's unfortunate, that I only was there because of a homicide, but it's a nice city. I looked up a couple of facts about it. So they average about 2.86 murders per year, which is not many, especially if you think about Indianapolis. If you think about the population, though, it's actually a little bit high, to be honest with you. So Michigan City does have a little bit of a higher homicide rate than other cities in the state. But still, when a homicide occurs in this location, it's uncommon. People take notice. And here's our timeline. So the defendant is John Hallett, pictured right there. And the homicide happened in 2017. So again, the victim is John Gonzalez. He's beaten and strangled in his home that he shared with Mr. Hallett. They were both formerly homeless people who were, through a social services agency, placed together as roommates in this house. And as I'll explain, one night everything went haywire and a murder occurred. So it wasn't until 2022 that he, just out of the blue, calls his local police department. He lived in Washington State at the time and said, "I killed this guy in Michigan City, Indiana in 2017." And so the police take these calls. I'm like, okay, so he came in and they did some interviewing and so on, trying to get some more details. He also called Michigan City Police Department and said, "I killed this guy in 2017." And so on. So both those police departments are, abuzz, trying to figure out like who is this person and if there's any truth to this. And ultimately, though, they are able to show, or they feel like they have enough evidence to say, yes, indeed, he actually did commit this homicide. Now, a year later, after some initial investigation and some development of evidence and so on, that's when they turned to us and they said, hey, we have these samples from the basement of this house. Can you see if they have these particular VOCs, these characteristic compounds that are also known as the scent of death? Okay. So the trial happened in 2024. I testified in that trial, and he was convicted and sentenced ultimately to 56 years in prison. He appealed that. Interestingly, the appeal was all about my testimony. He appealed whether the scent of death evidence was admissible. And that's your right as a defendant to challenge the admissibility of evidence against you. The trial judge said, yes, this is admissible. We even had a small, not a meeting, but it's a hearing called a Daubert hearing, where you actually go through point by point why the evidence should be admissible or not. So in any case, he appealed that, but they affirmed it. So they affirmed the conviction. And so that's essentially the end of the story for him. He's now in prison. All right. So in terms of the confession, we begin with why. It appears it's based on fear or paranoia. He thought that his roommate was stealing his mail and conspiring to get him evicted from the home in which they both live. And then the victim himself was older, was disabled, had muscular dystrophy, so was definitely a vulnerable, individual. And then the actual crime was okay. Oh, there we go. Sorry. He attacked him as he was coming out of the restroom in the house with a crutch. He beat him with the crutch until he stopped and the victim grabbed the crutch, whereupon he then beat him with his fist and then striked him. Okay, so very brutal. And then what happened is that he had, waylaid his roommate and he took him down to the basement and he left him there for about 56 days. So decomposition of a human body proceeds relatively quickly, especially if it's open air. And 56 days is plenty of enough time for a body to make its way all the way through all the stages of decomposition. Advanced decomposition is, well, the ultimate decomposition is skeletonization. There's basically no tissue left over. I'm not sure if it got that far, but basically that's a long time, for a body to sit there. So that means it's generating all sorts of decomposition products and fluids, and all of that is, accumulating there in the basement. He eventually dismembered the body and put it into the trash. And he must have done it well enough so nobody noticed. So it went out with the trash, and this body has never been found. Maybe someday, but up till now, no one has ever found him. And so that, if you might be familiar with criminal investigations, that's often a deal breaker. Homicide or proving homicide without a body is extremely difficult. And so it happened in this case, so we'll talk about that. But that's the problem, is that he chopped it up, threw it away, and then went to a fair amount of length or effort to try to clean up afterwards. So, mopping, he laid down several layers of gray paint on the concrete floor, all in an effort, to either remove or mask or, somehow exclude all that material. Now, no body, no problem. Remember, it's normally a deal breaker, and that's true. However, there have been exceptions to this rule, and so it is possible. There's actually a whole Wikipedia page dedicated to homicide trials without a body, and that means that you need something else. You obviously need additional evidence, and so you need either circumstantial evidence or forensic evidence to make up the difference. That you don't have the actual body there. The person you're looking at there is Kristen Smart. I don't know if you remember this case. She disappeared in 1996, and years passed, and they never found her body. However, the person that was with her the night that she disappeared, who ultimately got convicted, Paul Flores, they found at his father's home a plot of disturbed earth, and then they did chemical testing of that and were actually able to show that there was there once were human remains there. So they detected the presence of human blood and so on. And so that, along with some other things, were put together and actually played a role in convicting Flores of the murder. Okay, now, in terms of what they actually did, the very first thing they did is they used a canine. I've mentioned this already. We do a lot of research with canines as a forensic chemist at the IMHM. One of my jobs was actually helping to train and test canines to find explosives and ignitable liquids. It's incredible what canines can do. And one of the main reasons they can do what they do is that they are so sensitive. They have a sense of smell that's much, much more sensitive than ours. And so they brought in a dog that was trained to detect human remains. So it's called a cadaver dog, or human remains detection canine. And so in this case, they brought the dog down there and it alerted. Okay, so it showed a clear change of behavior. Said, I smell something down that smells just like decomposing people. Okay, so there's that. In addition, they noticed that in this basement, there's various seams in the concrete, cracks, those kinds of things. Those are the perfect places for liquids like decomposition fluid to collect. Okay, so it was not a perfect surface, and so that meant that there's hope, that there's might actually be some residual material still in there, and they can obviously, the dog found it, so then they hoped that they could find it too. Okay, so they looked at that and they said that we've got some potential fluids here. They did some presumptive testing for blood. There's a particular compound called luminol, and it's an amazing compound in that it will chemiluminesce in the presence of hemoglobin. So they use this product called Blue Star. That's just the name of the product, but it's the same basic principle. You spray that down, and then you turn the lights off, and then it glows. So literally, like, you see this blue chemiluminescence. And if you see that, that's an indicator, that you might have hemoglobin and hence blood, and hence, a dead body. So they did that and actually saw results. And then they took samples of the concrete. So this is what we ended up having in our laboratory, was circular samples about that big. And so they just drilled those right out of the floor, put them in packaging, and then it ended up in our laboratory in Indianapolis. So I noticed another important detail here is that DNA was not possible. I actually was asked this by, and it's not advancing, asked this by a member of the jury, why didn't you have DNA? It's not that they didn't try, it just wasn't possible. The samples were not suitable for DNA analysis. And so, can you remind me again what I do now? Oh, never mind. Okay. I just have to hit it all the time. All right, cool. Okay, so this picture is kind of neat. I'm going to show you the picture from the crime scene, but this is actually from the manufacturer. So it's just showing you a kitchen sink, lights out, all that blue glow, that's from hemoglobin. Okay, so if you're trying to clean up blood in a sink, something like that, that's what this is trying to simulate. And so the sensitivity is pretty ridiculous. Like, you could take blood, as it says here, and dilute it by a huge amount, 1 in 100,000, and it still will show up. And so that's one of the great things about it, is that it's so sensitive that even blood that's been washed or cleaned or wiped away or whatever, you can still see some of that residual. And it doesn't preclude DNA analysis. So this is the pictures from the scene. On the left, we've got one of the core samples, that's been drilled out. This is the hole left over. And then this is after they treated it with the Blue Star and turned the lights off. And you can see blue glow. Okay. Now, this is not definitive. This doesn't mean that there's blood and nothing else. There are possible false positives for this chemical reaction, but it's still a really strong indicator. And it gives you, motivation to take that evidence and do even more with it to try to see what you can see. All right, so let's talk about the set of death more explicitly. And so in order to do that, I want to define a couple of things. Let's talk about what's VOC. It's called a volatile organic compound. All that is in chemistry is a compound made from carbon that evaporates very easily. Okay, so, and instead of saying that, we say volatile organic compound. And so here's some examples there on the right. I can pretty much guarantee you that those are in the air in this room right now. Okay. There's VOCs all over the place, and they come from, basically any kind of object that's, might be in a particular location. So if you have anything like building materials, if you have any kind of paint, rubber glue, etc., if you have a bunch of orange peels lying around, then you would have, some of these compounds. I've listed their boiling point just, because I'm a geek, but also to get across the idea that all of these have relatively low boiling points. So they are easy to vaporize. It does not take much energy for them to become the vapor phase. Okay? So people care about these for a couple of reasons. Not many people think about the scent of death, admittedly, but like the US EPA cares a lot about these because of indoor air quality standards. And these can actually, if they're high enough concentration, they can make you sick. So you don't want extremely high levels of VOCs in your home and your workplace and whatever. And so that's why people have been studying this. This whole idea of looking at VOCs as a result of decomposition is a relatively new phenomenon. It's not, within the last year or so, but it's basically the last decade or so. People have been paying more and more attention to that. Some examples, let's start with some compounds that smell good, okay? So these are all different flowers, and what you're looking at here are the flowers like here on the top, and then these are all chemical structures. I'm not gonna belabor with what these structures are, but I just want to get the idea across that VOCs and aromas are completely linked. So the things that we smell with our sense of smell, that's because of the VOCs. So here's a bunch of different flowers, and you can see there's a whole diverse group of compounds. The number of VOCs is probably in the thousands easily. And of course, they differ depending on where they come from. So if it's a flower, you're going to get compounds that look like this. Okay, so and you'll notice too that aroma is almost always never simple. It's not like a single chemical VOC, it's multiple. So when you actually sense the aroma of something, you're sensing multiple chemical compounds all at the same time. And your brain is then translating that into the sensation of an aroma. So this is what you smell when you smell flowers. Inside your nose, there's a lot of stuff going on. If you look at this little cartoon, this is the idea of trying to get across the sense of smell and how it works. So we start down here. These are the VOCs. So odorant molecules or odor molecules. These are free floating. You breathe them in. And then inside your nose, you have first a series of receptors, a number of different types of receptors. And as I say here, they operate like on a key lock mechanism. So a particular type of molecule and its shape will lock into a particular receptor in its reciprocal shape and then bind. When that binding occurs, then the sense has been activated. Okay. And so we have lots and lots of these receptors. And there are, of course, different types to try to sense all the different types of possible molecules that could exist. And so from there, then it gets into our brain. And so all sorts of interesting stuff happens here, where we have olfactory receptor neurons that are linked explicitly to these receptors. Then it then gets translated or transmitted to structures in our brain like mitral cells and glomeruli. Those are described as way stations in this case, and they eventually transfer that information. So the sense of smell is a multiple step process. And it's pretty amazing that we have this built into ourselves. And it's over millions of years of evolution turned into this system where you can detect a wide array of compounds. And we as humans, we're not that bad. We have a decent sense of smell. We always, pale in comparison to something like a canine, but you kind of have to have a decent sense of smell because we use our sense of smell so much for a lot of different important things. So, and that includes, is this milk good or not? How many times have you done that? Where you sniff the milk, you are looking for sour. The scent of sour means acidic compounds. So things that build up in milk as it spoils are all acidic. To us, those smell sour. Same deal with other kinds of like sulfurous compounds or anything that's coming off of food. It's like spoiling and turning into mush in the back of our refrigerator. We detect potential mates. There's a whole segment of research on human pheromone recognition. And so they've done experiments where people recognize each other by smell. Illness. So if you've ever smelled someone's breath, if they're have a respiratory illness, you can actually detect the presence of that illness based on the smell. So there's all sorts of things going on here that are huge driving forces for our sense of smell to be highly attuned. Okay. So you may have heard this, but the sense of smell is definitely linked to taste. So if you lose your sense of smell, your sense of taste suffers a lot as well. During COVID, that was a pretty common phenomenon where people would get COVID and then they'd essentially lose their sense of smell. Some people temporarily, some people not, but then it would affect your sense of taste. Like you put food in your mouth and you get nothing out of it. So that's linked. Memory is another interesting one where we store some memories based on how it smelled. And so certain smells like, mom's home cooking can come back and will be transported back to those times. So we're pretty decent. Canines, of course, they've been doing this longer than we have, and they depend on their sense of smell even more than we do. And so, for a lot of reasons, including those, they're much better at it. And so it's, anywhere from 10,000 to 100,000 times more sensitive. Okay. Oops. So I mentioned death. When we die, chemistry continues. It's just the chemistry of life transitions into the chemistry of death. And so that means that during the process of decomposition, all the bacteria that are native to our bodies are running amok. Okay, and including new bacteria that might, supplant themselves onto us. And so that means that bacterial action generates a massive amount of VOCs, in particular methane, hydrogen sulfide, and ammonia. Okay. Now, they also generate side products, other configurations of molecules, and those become what we would term decomposition VOCs. So a whole bunch of other chemical compounds that are related to their starting material, whether it's protein or fat or whatever, and all of that becomes transitioned into a different form. So here's some examples. What I've got is a list of compounds according to chemical class. Now, the chemical class is not super important. That's just how chemists organize molecules. So we look at molecules, we look at the atoms and the bonds and the functional groups, and we say this is an amine, and we then classify them all as that type of compound. So, but basically on the left you've got the classification. Then I've got some examples, and then in quotes is the kind of gross part, but this is the qualitative descriptor of what this smells like. Okay, so some of them are a little bit on the nose, like cadaverine, hence cadaver smells, guess what, like decaying flesh. That's why it got its name, cadaverine. And so putrescine, same kind of deal, rotting meat. But then you have a bunch of other ones, the smell of vomit, rotten cabbage, musty, cheesy. Some of them in isolation would not necessarily be terrible, like phenol. Phenol is a decomposition of VOC, but it's also the same ingredient in those sprays that you spray onto your sore throats, and it numbs out your sore throat. So it obviously doesn't like smell like a dead body because no one would buy that, right? But if you have phenol mixed in with all the rest, then you end up with this very complicated picture that is unpleasant for us, but that is the basis. So that also means, from a chemistry point of view, these are the compounds we want to find. Okay, we're on the hunt for these because we know that these are formed as a result of decomposition. Yeah, has this happened before? Yes, it has. If you're up on your true crime, this is Casey Anthony, who was accused of killing her daughter, Kaylee, who's pictured right there. And they found her remains, so they did actually have a body in this case. And they tested the trunk of her car for VOCs, and they found VOCs in that location that are attributed to decomposition. So the thought was that she killed her daughter in some form or fashion, put her daughter into her trunk, and left her there for a certain amount of time, and then removed her, and then they had just VOCs left over. Ultimately, again, if you're up on your true crime, spoiler alert, she was acquitted of this. And so, who knows what actually happened. Like, for example, the defense actually made the counterclaim that she was transporting garbage in a truck. So if you have a bunch of garbage bags that leak, say, it's going to smell terrible. And so, maybe that's why they're able to detect those VOCs. I'm not going to, comment either way. Okay. So what did we do? This is our process. So we began, and here's Alexis here actually doing it. So there's a picture of her processing the pieces of concrete by trying to remove the VOCs or the chemical compounds from the concrete and get them into an instrument that can make a measurement. So the instrument that we're using here is called a gas chromatograph mass spectrometer, or GCMS for short. You may have even heard this mentioned on TV. It's a pretty popular instrument. It also is fantastic at analyzing things that are volatile and giving you a pretty good idea of what their identification is. So we started with just looking at it, taking photos, scraping debris off the top, and then using just swabs. So fresh, clean swabs, not like you get, Q-tip swabs, but like, lab-based swabs. So they're like $5 a piece. And so then you have to just rub it, on the surface. And you have a wet swab first, and then you follow up with a dry swab. Then all of that gets put into vials and then analyzed. Luckily we have a robot that does all of that work for us. So once you have samples in vials, it'll automatically go along and heat the sample and try to determine if there's any VOCs present. Then ultimately, you try to identify everything. You identify those by using a mass spectrometer. A mass spectrometer is a really powerful instrument that allows you to look at a molecule as well as the pieces that it generates under controlled conditions. And those are essentially considered like a fingerprint, quote unquote. Okay, so it's considered to be a pretty definitive identification. All right, so the first thing that we did, the method that actually captured the VOCs is called solid phase micro extraction. Okay, it works like this, where you basically have what looks like just a normal syringe. If you extend the needle of the syringe, at the very end of that needle is a coating, and that chemical coating is there to absorb things from the air. So you basically put your sample into an airtight vial. You pierce the septum on the top of the vial with this needle, expose the fiber, wait, retract the fiber, and then you take that same needle and bring it over to your instrument, the gas chromatograph. So in that case, you put it into the gas chromatograph, you expose it. The heat inside of that instrument is enough, so whatever's on that fiber comes off, it gets released. And then it continues on into the instrument, and then you can analyze it. So solid phase microextraction is a very popular technique in analytical chemistry. It's super fast, easy, sensitive. And so it's basically ideal, for these purposes. And we use it for a whole wide array of things. So we actually have all the right equipment to do this kind of experiment because we're constantly being interested in what kind of volatile compounds might be. And the next step, the important part, is actually identifying things. And so the GCMS, gas chromatography mass spec, it's a two-stage process. So if you look at this little diagram here, if you look at the box in the middle, this is just an oven. And inside the oven is a long, thin glass tube. It's called a capillary column. And so what you do is you take your steamy fiber, you stick it into this heated inlet, and all those chemical compounds come off of the fiber. And then they enter this column and it's being pushed continuously by a flow of gas. And so the chemical compounds actually transit this column. They make their way through all the coils of this column and eventually come out at the end. The secret is that every compound that you put in there has a different velocity. Some go slow, some go fast. And so that means if you have a mixture at the beginning and they all take their own time, to go through this column, then at the end, they're coming out one at a time instead of all at once. And so if they're coming out one at a time like that, that enables you to use this part of the instrument, the mass spec, to actually ionize that molecule. It breaks apart, it measures the mass, with everything. You can search it against the database. And basically you can do this one experiment, it takes about 30 minutes. And you can say, okay, I've got, 6 different chemical compounds in the sample. Compound 1 is this one, compound 2 is that one, and so on. And so it's a pretty powerful technique. And so, especially because it combines a separation step, the GC part, with an identification step, which is the mass spec. So let's show some data. I'm going to show you just a couple of the samples. We did 21 samples on all, so I'm not going to sit here and show you all 21, but I want to show you just some examples. So on the left, we have item number 4. And in this case, this pink stuff, that's from Blue Star. When you rub something with Blue Star and turn the lights off and so on, it leaves behind a stain. Okay, so you see this purple stuff. So that's normal. That was not there when they first gathered this thing from the floor, but it's also okay, doesn't interfere with it either. Okay, so that's that sample. And in this case, there's peaks. So every one of these vertical spikes is a separate chemical compound coming off of the column one at a time. Now, what we can do, of course, is identify all of these and say, are any of these something that we have not seen in our control samples? And are they decomposition VOCs? So in the case of this, yes, we're seeing compounds, but no, none of these are known to come about by human decomposition. So this is a negative. And so the number of peaks that you see, it all depends on how complex the sample is. But basically, the separation technique is such that it can handle pretty complicated samples. So here's another one. This is also negative. So just a piece of the concrete. Every one of these peaks has been identified, but we can say no, none of these are known to be human decomposition products, and they are also not in our control or in our control. All right. Now that we have this one was labeled a control. A control in a scientific experiment is a piece of material that you analyze that you expect to be negative. Okay, so it's a check, essentially. However, we learned kind of an interesting lesson that a control in this case doesn't have a ton of meaning, because what this is a piece of the concrete floor across the room from where all of those other samples were taken, and I'll show you what those are. So these actually, or this one actually shows VOCs. So peak 1 and 2 are, if you look it up in the literature, they're known to come from decomposition. So this is just one of those chemical compounds that we form when we die. Now, why is it something that's quote unquote a control? This is another thing about VOCs is they move. Smells migrate. You know this. And so if we're looking in the same room over many years, VOCs can diffuse. And so it wasn't at first it was confusing, but then it started to be less of a shock that we might see this. So we're actually seeing degradation of VOCs here. And then we saw a whole bunch here. So this is item 10, and all of these compounds, again, are known to occur as a result of decomposition. Now, an important point, when you do these experiments, a lot of people use humans, like body farms and stuff like that, but a lot of people use animals, like pigs. Right now, the state of the art is we cannot tell the difference. And this is an important thing for me to tell the jury on the stand, is that I can tell you that these are decomposition products of something decomposed here, but I couldn't tell you who, and I can't even tell you whether it was a human or not. Okay, so there's an important limitation here. And that's the kind of the way it goes with forensic science. You have to be able to talk about your evidence and give it its due, but do not oversell it. Do not say something about the evidence that's overly optimistic. So, but in any case, we have a whole bunch of VOCs showing up here, and those are all nicely highlighted there. And then we brought it all together. Like I said, we had a lot of samples. And so this table just lists all the different VOCs that we found in that basement. Lots of them on lots of different things. And so this, as I just said, human versus animal is still a point of contention among people that are doing research on VOCs. So in the literature, you can see these compounds being formed by humans or pigs. Okay, both of them occur. And so you can list this all the way down. And so, but taken together, the more we looked, the more we found. And so these VOCs just kept on popping up to the point where we came up with this wording in the report. So basically, taken together, we have 4 different items, have 9 different VOCs that are known to come from decomposition. So that leads to the conclusion that the decomposing body of someone or something, importantly, was present in the room for enough time for the decomposition of VOCs to collect on, in, and under the floor. And the fact that this works, that we saw these and we can confirm them, even after years and after a huge amount of cleanup efforts, including painting, which actually ironically probably worked against him. If you paint, you seal up VOCs. So in this case, though, it was all there. And so this is clearly not, definitive that absolutely there was that person's body in the basement, but it's circumstantial evidence. And a lot of forensic evidence is essentially categorized as circumstantial. It indicates something, but it can't, tell you with 100% certainty. Okay. So some pluses and minuses for this. It's great because they are sticky. They go everywhere. They persist. And so even after years, they'll stick to a surface and then you can find them again. And so that's great. That obviously helps confirm the presence of something decomposing in that place. Of course, the limitations, though, they're not specific. They're not like DNA. And again, I actually even got a question from a juror during the trial whether or not this is like DNA. The answer is no, it's not. And then there's the issue of human versus animal. And so right now we don't know the difference, or we can't reliably tell the difference between the two. Okay, so then lastly, why does this all matter? I wanted to just broaden this out. I put this together based on just personal knowledge and also research that we do. If you look at VOCs, they are all over the place, and they're used by both instruments and canines to indicate the presence of a number of different things. So all the stuff you see around the edge, you've got human remains, which was the whole topic of this presentation, but also explosives again, which is something that we've worked on, but drugs, the presence of illness. If you've ever heard of the Beagle Brigade, they detect fruits and vegetables at airports. And so they do that based on VOCs. Believe it or not, there are USB drive canines. So you can actually train a dog to find electronics of different types. Bedbug dogs, so you can find the odor of the pests. And so on, and then of course ignitable liquids like an arson scene. So the ability to detect something by VOCs is pretty powerful because number one, we have really sensitive ways to do it, but two, they're really strong indicators. And so, and they're always there, they got to be persisting there in the environment. So then the last thing I wanted to show you is actually, this is a canine detecting explosives, but I just think it's a fascinating video. It's one of my favorites. So I wanted to show you what it looks like, what a canine actually does do an alert. This is a canine from the UK, and it's being trained, or it's trained to operate off lead. Normally you have a leash, in this case you don't. And it's going to run down, and there's a whole bunch of identical containers at the end of this range here. One of them contains about 100 grams of a high explosive. Okay, so I'm going to let you watch this. I think it's going to be pretty obvious which one it is. Let me play this. Guess what? I think it's the number 4. So it's really impressive what they can do. And of course, they're operating on the same principle. They're looking for VOCs just like we did, just we were, doing it in the laboratory. So I just want to say thanks very much. And that concludes the talk.